Let's go ahead and get started. We only have 3 lectures left, and you guys do the last 2 lectures. They're poster sessions, right? So we're almost done, but so we'll have to be streamlined a little bit, but we'll just go as far as we need to go, and we're not adding more material or anything like that. So the quiz, of course, is on Thursday. Usual things apply. It's the last group test that you'll take, so make sure you take advantage of that. Any questions about quiz 3 material? We're all on board with that? Okay. Sorry, unfortunately this lecture was not posted or revealed or whatever it was, so I did that this morning. Probably too late for you to download it, but it is up there now. We have just a little bit to finish on lecture 17. We're talking about G proteins, and then we're going to pick up one more type of G protein, and then move on to the receptor tyrosine kinase growth factor receptors, which are really important in the field of cancer. But before we do that, so we've got a lot of different tools that we can use to modulate this cyclic AMP signal that we've been talking about, or cyclic. We shouldn't limit it to cyclic AMP, it could be cyclic GMP, either one of those 2 nucleotides. One way you turn those things off is you use phosphodiesterase. Cyclic AMP phosphodiesterase for cyclic AMP destroys the signal, and cyclic GMP phosphodiesterase destroys cyclic GMP, and you have inhibitors like caffeine for the cyclic AMP phosphodiesterase, and then Viagra and Cialis is for cyclic GMP phosphodiesterase. So these are both inhibitors of the destructor. So if you inhibit the destructor, what happens to the signal? It stays high, right? So in both cases, if you drink a lot of caffeine, then you have an elevated cyclic AMP signal. And of course, if you take Viagra, you have an erection because, or some of us would, because it inhibits the phosphodiesterase that destroys the signal. And so therefore the signal stays up longer. What happens if you take Cialis? You sit in the bathtub and look at the sunset. That's what the commercials say. Okay, so a couple other things. Let's look at these toxins down here, because we need to pick up 2 important tools. And to go through these, first of all, I'm going to tell you what they do, and then we're going to use it in sort of a problem. So again, both of these toxins are going to affect cyclic AMP. And the reason they're going to do that is, so what cholera toxin does is it takes this weird molecule called ADP ribose, and there's no reason to know the structure. It's just a sugar molecule attached to a nucleotide, right? And so it takes that structure, and so it's when it attaches it to an arginine residue on the G sub S alpha, what happens is it prevents the GTPase from turning on. Okay? So if you ribosylate, cholera toxin will ribosylate the G sub alpha, particularly in the intestine. So that's usually where that toxin is encountered. You have contaminated drinking water, and so the cholera toxin then ribosylates the GS sub alpha and prevents the GTPase. What's that going to do to your cyclic AMP levels? What would it do? So we've got this situation. Let's just draw it out. Let's pretend we don't know what's going on in the intestine, even though we do. So here's the cell membrane. You could put, let me turn this on. Okay. So this is adenylyl cyclase, and we know what's going on here. You have ATP, and so you run it through this little enzyme. If you activate it, and that creates cyclic AMP, right? So we've already talked about receptors like the beta adrenergic receptors. Adrenergic receptor. That would be this guy. That binds to something like epinephrine. Okay? So epinephrine binds to that, and that's coupled to, let's put it in green since it's stimulatory. So this is a GS alpha, okay? And we know that when you activate GS sub alpha, it stimulates adenylyl cyclase. So if we put cholera toxin on here, and so that cholera toxin is going to ribosylate this guy right here, particularly when it's stimulated, okay? And so it's already bound to GTP, isn't it, in the on state. And so it will stay on because cholera toxin is going to cause that to be incapable of breaking down its GTP, and therefore it's always stuck in the on state, and that will continually stimulate adenylyl cyclase, and so we'll get a what? An increase in cyclic AMP. Right? And what happens is that increase in cyclic AMP causes increased secretion of water and salts into the gut fluid. And that's what creates diarrhea, intractable diarrhea. And people die from cholera not because of the cyclic AMP, but because of the dehydration of all that water going through. Now, in the old days, what they used to do is, if you came into a contaminated region like what happened in Haiti, and it just happens all over the place. People still die from this. Millions of people die. And so the problem was they used to give people IV fluid, right, to replace the water that they lost. And that keeps them alive long enough for the antibiotics to kick in. But that's really expensive to give someone IV fluids in this sort of situation where there's a lot of contamination. What's something else you could do? Well, you know about the SGLT1, right? Now, if you give sugar water to those same people, what happens is the glucose and the sodium are taken in through SGLT1, and then they are absorbed into the bloodstream, and so they oppose the secretion. You're releasing fluids, but at the same time, you're taking them back up because the person's drinking clean sugar water. And let me tell you, sugar water is a lot cheaper than IV fluid. And so that keeps them alive, again, long enough to get some sort of antibiotics going to kill off the bacteria to shut this problem down. Okay. But if you didn't know, so let's pretend now we don't know what cholera toxin does, right? Or we don't know that what's causing the fluid flow. So let's forget about, we don't know the receptor, and we don't know which G protein is involved. How could you use cholera toxin as a probe? Okay. Well, the first thing, let's pretend we know one thing, and that an increase in cyclic AMP stimulates this flow of fluid, right? So how can you stimulate adenylate cyclase without using any receptors or hormones? I've given you a tool from lecture 17. There's a plant drug that directly activates adenylate cyclase. Okay, so to confirm that cyclic AMP is involved in this process without worrying about the type of G proteins, you first put on forskolin. Forskolin is a plant drug that stimulates this, okay? And if the fluid flow is caused by cyclic AMP going up, then you'll get fluid flow, right? You'll get this salted water secretion into the gut lumen. So now the question becomes, okay, it's due to an increase in cyclic AMP, and if you really want to be fastidious about this, you first show that if you destroy the cyclic AMP signal, that prevents the fluid flow. How can you destroy the cyclic AMP signal? One step above that. You don't give them Viagra, you give them you give them caffeine. That's going to elevate the signal, right? So that caffeine should elevate it, right? So that's another way of stimulating cyclic AMP. But what you need to do is you need an inhibitor of the cyclic AMP phosphodiesterase, some sort of inhibitor. Okay? So that would break it down, and you would show that stopped fluid flow. If you cut this signal in two by using cyclic AMP phosphodiesterase, then that should diminish fluid flow. Okay. Now, if we want to pin it down, and so we put epinephrine on there, all right, and epinephrine stimulates fluid flow, fluid secretion. But you want to ask the question, which type of G protein is being stimulated by epinephrine? Let's pretend we don't know. If we put cholera toxin on there, that should stimulate adenylate cyclase, okay? And so that will help us identify that, in fact, G sub alpha or G sub S is actually involved in this process. So if you put cholera toxin on there and it stimulates it, it shows that there's a G sub alpha, GS sub alpha that's involved in the stimulant. Well, what if there was an inhibitor? Let's look at this situation. Now, because in many situations, what you have is you have a stimulatory G protein, and you also have a G sub I, which we talked about. Okay? And the role of G sub I is to balance the stimulation by G sub S. Right? So you can have both of these. And in fact, this is the situation that you have in the airways. Okay? In the airways. And have we talked about a receptor that activates G sub alpha, a G sub I? Do you remember what that one is? Is in the heart. Acetylcholine is the messenger. Okay, it's got to bind to a cholinergic receptor, and which flavor of cholinergic receptor? What are our choices? Nicotinic, could be nicotinic, but that's not what's going to go on here because that would open a sodium channel. We want to activate muscarinic. It is specific flavor of muscarinic. It's M2. Okay? So that's the muscarinic, it's the nicotinic muscarinic M2 receptor. Okay? And we know that's coupled, what that does is it stimulates G sub I, and what that does is the effect of G sub I on adenylate cyclase is inhibitory. So that's going to decrease cyclic AMP, right?
That's in the heart, right?
I'm sorry?
That's in the heart.
No, we're in the airway now. We're in the airway. And this system is actually in the airway, both of them, some sort of a push and a pull. And so, why would you want to stimulate fluid secretion in the airway? Think about cystic fibrosis. All the mucus, right? But you can overstimulate. So this is really carefully regulated. You don't want people to have cystic fibrosis, so you want to have fluid flow in. So this is the airway now, okay? All right. You want to have fluid flow so that it rinses the mucus and all that stuff out. But if you produce too much fluid, what do? Somebody just did that. You hear that cough? Okay. That causes people to cough, right? Is when you get a little bit too much, and you're trying to clear your throat and get rid of all that stuff, right? But if you do this all the time, you can get what's called whooping cough. And that's actually, we thought we had extinguished it, but whooping cough has made an appearance in certain places in the United States where parents were afraid of inoculating or vaccinating their children because they thought it caused autism, and it doesn't. Vaccination does not cause autism, right? And so these very well-meaning, very wealthy people that read too much internet news thought that autism was caused in their children by getting vaccination, so they didn't vaccinate them. So guess what? They came down with these weird things that we all thought we were protected against, like whooping cough. Okay, whooping cough, let's talk about what causes whooping cough. And it's down here, it's in your notes. So pertussis toxin is what is the active ingredient of that bacteria. And so pertussis toxin, it also ribosylates a G protein, but not just any G protein, only GI, the inhibitory G protein. Okay? And again, it ribosylates the alpha, but it will only ribosylate the alpha subunit if it's bound to the beta gamma subunit. And if it's bound to the beta gamma subunit, what is the GTP state of the alpha subunit? Is it bound to GDP or GTP? It's bound to the beta gamma tightly. It's bound to GDP. It's in an off state. So pertussis toxin is very different from cholera toxin because so this is pertussis toxin. Again, it ribosylates this, but it inhibits it. And the reason it inhibits it, unlike the G sub S, is that it prevents the G sub I from breaking apart and losing the GDP, picking up GTP. It prevents GTP exchange. So that means you've inactivated the off switch in the lung, right? So the thing that's left on is the on switch. The G sub S is still there, churning away, but now you have no way to turn it down, right? So what happens is you get all this fluid flow all the time, and the person starts to cough, and that's what the whooping cough is. Okay. But you can kill it with, antibacterials. Okay, so remember how to use this as a tool. So if I give you a situation where, is this driven by cyclo-KMP? The first step is not to start putting hormones on there. It's to prove that it's cyclo-KMP driven. So you put forskolin on there. If it doesn't do anything, you don't have a cyclo-KMP driven system. Okay? So then the next question becomes, is it a G sub alpha or G sub S or G sub I? Or maybe they're both there. Is it sensitive to cholera toxin? In other words, does cholera toxin stimulate it? If it does, the answer to your question is that G protein is involved. Okay. If you throw on pertussis toxin and it doesn't do anything, then this thing isn't involved. Okay. But if you throw on pertussis toxin and it decreases, you actually get an increase in cyclo-KMP, what happens is this has been inactivated and you do have a G sub I acting in your system. So the activity of both of these is going to do the same thing intracellularly. The activity of both toxins will elevate cyclo-KMP. Both of them. For different reasons. Okay. Any questions about that? You've got all these tools to be able to manipulate the system. And now what we need to do is pick up a third major category of G protein, and that's the G sub Q family, where we're going to manipulate calcium levels. Okay? All right. And so that brings us into lecture 18. And we start to use a different set of second messengers. So let's pull this thing down. We're done with that. And we're into lecture 18. Okay, so we've been talking about different second messengers. So now we're going to go to a new category of second. These are lipid-derived second messengers, but there's not a lot of new things to it. We've actually already talked about where these lipids come from. These are in the cytoplasmic leaflet. These lipid messengers are related to this lipid molecule in the cytoplasmic leaflet. It's the head group is an inositol, so it's a phosphatidyl phospholipid, okay? And so we're going to do something to use this and turn it into a second messenger. So how are we going to do that? Okay, so the category of G protein that we're going to talk about is this right here, G sub Q. So it activates some membrane protein that's associated with the cytoplasmic leaflet. And that enzyme is called phospholipase C beta. It's this really long, ugly name. And it's important that you know that isoform right there, okay? Because the beta isoform is very different from its cousin, which is the gamma isoform, and we'll talk about both of those. So if it's a lipase, that means it's going to split the lipid molecule in two, okay? So this just shows you how you create phosphatidylinositol. So you have this phosphatidylinositol molecule that's unphosphorylated. And so a couple of lipid kinases, so these are not protein kinases, these are lipid kinases. So phosphatidylinositol kinase puts a phosphate group onto very particular places. Okay, a really typical molecule that's created, if you do it twice, is at the 4 and the 5 position. Okay, we've talked about this. This is the binding partner of AP2 and AP1, right? The alpha adaptive binds to PIP2. That's PIP2. That's just a sort of a shorthand name for it. PIP2 specifically refers to phosphatidylinositol 4,5-bisphosphate. So that just means that it's got 2 phosphate groups attached to the head group. Is this thing negatively charged? Absolutely. It's bristling with negative charges. So proteins readily recognize that. One of those proteins is the alpha-2 adaptive, right? That we've already talked about. In this case, we're gonna cut it, right? And when we activate the G sub Q, so we have some receptor, and the receptor that does this, a good example, is again, the ligand is acetylcholine. It binds to a muscarinic receptor, not a nicotinic. But in this case, it's called the M3, the muscarinic 3 receptor. And that receptor activates G sub Q. All right? And you know the story, so the G sub Q breaks apart into alpha and beta gamma, and let's ignore the beta gamma. It doesn't really do much in this situation. It's the alpha subunit. And what it's going to do is it's going to activate that phospholipase C beta. And that's going to cut PIP2 in a very particular spot, creating 2 different second messengers. One is called DAG, diacylglycerol, because it no longer has a head group. It's just glycerol with 2 fatty acid tails. That has to stay in the membrane. And there's a reason it needs to be there. So we'll talk about that, all right? So this half of the second message stays in the cytosolic leaflet. The head group then becomes the second messenger. We have a name for it. It's called IP3. If you want the technical name, it's inositol 1,4,5-trisphosphate, but the shorthand is IP3. Okay? This is highly soluble, so it's extremely happy to go flying around the cytoplasm looking for a binding partner. What is its binding partner? Its binding partner is a protein in the endoplasmic reticulum that's actually going to release a puff of calcium when IP3 binds to it. So the IP3 receptor is a calcium channel in the smooth endoplasmic reticulum. And you know what is going on in the endoplasmic reticulum. Remember, Circa has been packing all that calcium, removing it, constantly cleaning it out of the cytoplasm, and storing it in the endoplasmic reticulum. Okay? What's going to happen so here's the IP3, so here's that system. So we can start all the way back up here. You put acetylcholine, this is the M3 muscarinic receptor. It activates a G sub Q alpha protein. That alpha protein, this thing right here, activates the enzyme which cuts DAG away from IP3. DAG stays in the membrane. The IP3 then drops into the cytosol and then binds to the IP3 receptor, which is what's called a calcium-induced calcium release channel. Okay? Calcium-induced, that's very important actually. CICR, calcium-induced calcium release channel. Okay? There's two ways to open this channel. The first is simply by binding IP3 to it. That gates the channel. So this is what type of channel? It's a ligand-gated, it's an intracellular ligand-gated channel. It opens, which way is calcium going to move? It has to go into the cytosol, right? You're going to get a spike, is really what you're going to get, okay? Now, what happens is you don't need very much of this. In fact, just a little bit of this is going to produce a little smudge of that. You're going to open one of these IP3 receptors. What happens next is this little puff of calcium actually will go next door and open other IP3 receptors. So there's two ways to gate this channel. It is gated by a ligand, but it's also gated by calcium. So it's an ion-gated ion channel. And so what you do is you get a concerted wave of calcium being released, because this one activates its neighbor. It's kind of almost like an action potential. So you're getting this wave of little calcium channels opening and opening and opening. And you don't need a lot of IP3 because the calcium itself will open its neighbor. And then that will open its neighbor, and that will open its neighbor. So you get this wave of calcium blowing through the cytosol. What does it do? Calcium is a second messenger. So we got lots of second messengers. We got DAG, we got IP3, we got calcium. What's so important about calcium? All right, so calcium is a really good signal because the baseline of calcium levels in the cell are so low. So all you need is a little puff, and the cell will immediately respond to it and actually start getting rid of it as soon as it can. But before it does, it's going to activate this kinase, protein kinase C. It's called protein kinase C because it binds to calcium. That's where it got its name. Okay? Now, protein kinase C is soluble in the cytosol. Okay? It has a domain that's called the C2 domain. Okay? And what happens is calcium binds to that domain. So here we are talking about PKC. And there's lots of different isoforms of PKC. But what happens is if calcium binds to this, it's going to traffic to the cell membrane and bind to DAG. How does it know to do that? Well, it has a helper that your book doesn't talk about a lot. Okay, it's called a RAC. And RAC is a funny name for receptor for activated C kinase. That's what a RAC is. It's not IRAC, it's RAC. Okay. So RAC binds to the C2 domain of protein kinase C if and only if it is bound to calcium. So calcium binds to PKC, it's then recognized by RAC, and then RAC drags it and targets it to the plasma membrane. That's why you have to have RAC. It's a targeting device. And it will take it, and different isoforms go different places, but we're only going to look at this one instance here where PKC is drugged to the plasma membrane. And when it gets there, if and only if there's lots of DAG in the plasma membrane, then PKC becomes fully active. So you see it has to have two signaling pathways to activate that. That's why we call protein kinase C a coincidence circuit. This is a coincidence gate, actually. Right? If you have something really dangerous like PKC, PKC is extremely carcinogenic. Okay? You want it tightly regulated. You have to have this signaling pathway activated, and you have to have this signaling pathway activated. And then, when both of those converge at the plasma membrane, PKC can be fully activated, and it turns on transcription. Right? It's going to turn on all kinds of genes that lead to what? Cell proliferation, for one thing. Okay, so it's very dangerous, but it's very useful. If you want to divide cells, you put a little PKC in there, and that's what happens. It also does some clinically some dangerous stuff that we'll talk about a little bit later when we talk about the insulin receptor. Okay, so are you comfortable with this signaling pathway? All right, so we talked about how you get the spike But a spike isn't a spike unless you can turn it off. So if it just blobs out there in the cell, what's going to happen is you'll get all the good stuff. This will become active, but then the cell will kill itself. If you leave the calcium signal on too long, that's a death signal. Okay? So what you want to do is drive it down as soon as you've created it. So how can we turn off the signal? You know about some things, right? What would turn off the calcium signal? How can we get rid of the calcium? We're going to have to use transporters of some sort, right? Circa, because that's a really good one. That's right there. So it's going to start. Circa's slow, but it's just always working to pack the calcium back in there. So Circa will help drive this down. Could we put a Circa-like pump on the plasma membrane? We could put one on the plasma membrane. You can expel it. That's a quick way to get rid of it. Okay. So we talked about. Let's use pictures instead of words here. We'll come back to that signaling pathway. So we're talking about how to create the spike. We know how to make the spike go up. There's actually, what are the ways can you cause the signal to turn on? There are obviously calcium channels on the plasma membrane. We talked about one of those last time. It's activated by cyclic AMP, right? Epinephrine causes cyclic AMP to go up. Cyclic AMP turns on PKA. It phosphorylates a calcium channel. The calcium channel opens. Calcium always goes into the cell when you open a calcium channel. So that's another way to add to the peak. And there's also something called a store-operated channel. Okay. So these are two additional ways that you can create the peak without even using Circa. So we've got several ways of elevating calcium. So then we need several ways of destroying the signal. And we started talking about that already. So you can put a calcium pump on the plasma membrane. You can activate Circa. You can activate the sodium calcium exchanger. That's always at the cell surface. So that's getting rid of calcium. Okay. What else could you do? Could you use something to soak it up? Yeah. If it's bound, it's not active. Okay. So you can use calcium binding molecules. And so there's several. There's one in the ER. It's called calsequestrin. Okay. That's a calcium sponge. But there's also one in the cytoplasm. It's called, strangely enough, calbindin. It's really unimaginable. Calbindin. So it's a calcium sponge. That will quickly extinguish the calcium signal. Okay. And finally, we could use this guy right here, the mitochondria. Although we didn't explicitly talk about it, you should understand that this is extremely negative in here. Okay. That's part of the proton motive force, right? It's actually going to pull protons back through here and generate ATP. But you can do a lot of things with that proton motive force. Besides that, you can suck calcium into the mitochondrial matrix and sequester it. So that's a cleanup, right? Cleanup. And it's good because when calcium enters the mitochondrial matrix, it stimulates the oxidative phosphorylation genes to turn on. And so that stimulates ATP production, right? So that's good. You're getting 2 for 1. You store calcium and you stimulate oxidative phosphorylation. All right, so we have numbers of ways to increase the spike and numbers of ways to make it nice and sharp and get rid of it. So calcium is a dangerous signal, and one of the reasons for that is because, going back to PKC, let's go back and look at particularly what it does. So there's many isoforms. A lot of them activate protein kinases. Okay, and it turns on gene transcription. It actually will help turn its own signal off. It will activate that calcium pump at the plasma membrane. It will activate the sodium calcium antiport. So it activates the pathway to get rid of its own signal. So that's sort of a negative feedback, right? This is such a dangerous signal. Not only do you need a coincidence circuit to turn it on, it's actually helping to turn itself off. And the other thing it does is it phosphorylates numerous G protein coupled receptors. What happens when you phosphorylate the cytoplasmic domain of those receptors? You remember what happens? It can't, so if you serine, so PKC, remember the first question you ought to ask about a protein kinase is what does it phosphorylate? It phosphorylates serines or threonines, okay? And if you phosphorylate serines or threonines, as we'll see here in a moment, that makes it unable to bind to a G protein, so you effectively shut down that receptor. So this is also an off switch. And unfortunately, as I mentioned earlier, it also phosphorylates insulin receptors and desensitizes them. That's actually bad. And we'll talk about the relationship to type 2 diabetes here in a little bit. Okay, so you've released this calcium signal. One of the things that is the effector of the calcium signal, besides PKC, is calmodulin, right? And this is a protein that, let's go back here, we kind of blew past this. We need calmodulin to actually finish interpreting this little piece right here, okay? This is that, relaxation of vascular smooth muscle. Remember, it was sort of this your book gave you sort of a hand-waving explanation. Acetylcholine bound to something, and then there were a lot of arrows, and those arrows somehow activated the nitric oxide synthase system, okay? Well, what we want to do is clean up this little piece right here. What you should now know is that if you put an M3 receptor here and acetylcholine is dumped on top of it, what it will do is activate the G sub Q. And the G sub Q will then do what? It will activate this cascade right here. It will activate phospholipase C beta, okay? That will split PIP2 into two pieces, DAG and IP3. IP3 will then go to the ER, release calcium, and then calcium doesn't do anything by itself. What it does do, though, is it binds to a protein called calmodulin. Calmodulin is one of the effector proteins that recognizes the calcium signal and turns it into an action. Okay. So the calcium calmodulin complex is what binds to nitric oxide synthase and turns on the production of nitric oxide. So now all those little arrows mean something. So you have the rest of the story, right? And so it produces nitric oxide. This is produced in the endothelium. It goes next door, activates guanylyl cyclase. You get cyclic GMP, activates protein kinase G, and you get smooth muscle relaxation. Right, okay, good. So let's go back. Calmodulin is an interesting protein because it undergoes dramatic shape changes under two conditions. Number one is if it's bound to 4 different calcium ions, it's fully loaded. And so it looks like this, okay? That means it's ready, it's active to bind to its target. And one of its targets, of course, is nitric oxide synthase. But in this case, it can actually activate a whole set of serine threonine enzyme protein kinases that are called, strangely enough, calmodulin-dependent kinases. Okay? And so when it encounters its target kinase, what it does is it undergoes this dramatic conformational change and wraps around that protein so that this N-terminus is very close to the C-terminus and activates that kinase. And so this thing can go around phosphorylating transcription factors and doing all kinds of interesting things. Okay? One of the interesting things it does is actually in the central nervous system. It actually forms the basis for some types of memory. Okay? How can a protein give you memory? Okay? Well, we're not going to go into the details of the neurobiology, but what I am going to show you, so this is calmodulin kinase 2, and that isoform is found in a lot of neurons. Okay? And a lot of kinases have an auto-inhibitory domain where that catalytic domain is actually bound to this inhibitory domain. It basically has its foot in its mouth. Okay? So to activate this kinase, you have to do something extraordinary, and that is pull the foot out of your mouth. Right? And so what happens is when calmodulin detects a puff of calcium, calcium calmodulin is ready to go. It's looking for binding partners. Okay? And then when it encounters this CAM kinase 2, it gloms onto what? It binds to the inhibitory domain and boots out the kinase domain, so this thing becomes active. Partially. Okay? So it's partially active, and then what happens is, this is a kinase, but the first target that it activates is itself. It will actually, the kinase will phosphorylate that auto-inhibitory domain in activating it even more. So it becomes fully activated when two things happen. Calmodulin is wrapped around it, and when it has autophosphorylated itself. So it can undergo partial or complete activation. So after a while, it phosphorylates proteins until it moves proteins around that cause changes in synaptic formation and create memories for you and me, right? And after, very quickly though, you get rid of the calcium signals. You think, oh, well, you never would remember anything because you don't have enough time to really do much. Well, what happens is, this is a memory protein. What happens is, after the calcium signal dies off, calcium is ejected, calmodulin is ejected, but because it's self-phosphorylated, that's a form of memory. It retains the trace that it was activated earlier in the signaling pathway. So it remains partially on, and this is a form of molecular memory. And so it can continue to phosphorylate and activate proteins that construct synaptic junctions and memories, basically. Okay, so this is just another really good example of an effector for the calcium signal. Okay. And we've talked about these other things. And of course, if you want to, one of the things is these pathways that I've mentioned to you before talk to each other. It's called, oddly enough, called crosstalk. Can you have the cyclic AMP pathway talk to the calcium pathway? Absolutely. Okay. One of the ways you can do that is PKC, when it's activated, not only phosphorylates the muscarinic receptor, it will actually phosphorylate the adrenergic receptor too, and so it will turn off the other pathway as well. So that's a general off signal that PKC is doing. So that's crosstalk between the calcium signaling pathway, and if you phosphorylate the other guy's receptor, which in that case would be the adrenergic receptor that creates the cyclic AMP signal, if you phosphorylate that and turn it off, you're talking to the other pathway. The other way you can do that is calmodulin will activate a cyclic AMP phosphodiesterase. Is that going to turn off or on the cyclic AMP pathway? If you break that phosphodiester bridge, you turn it off. That's what the calcium signal is doing, is it's turning off in two different ways the cyclic AMP pathway. And it actually turns itself off because it extinguishes the calcium signal, and it also phosphorylates its own receptor, which would be the M3 receptor. So it's self-controlling, but it's also talking to other pathways. So that's just one of those examples of crosstalk. Okay. We've already talked about this issue about some proteins regulate ion channels, and we use the vagus nerve and acetylcholine as a way of activating the G sub I. And remember, don't be so naive as to expect that only the G sub I alpha is doing everything. In this case, the G sub I beta gamma also opens a channel which actually helps inhibit the muscle cell. We've already talked about that. Okay. Now, let's talk about, in general, the problem of G protein coupled desensitization. All right? This is a really big issue because you cannot leave these receptors on all the time. They're too dangerous. Okay? And when you do leave them on all the time, like they get a mutation, what kind of mutation would leave on a G sub S? Let's say you're going to mutate it somewhere. You're not ribosylating it, you're going to mutate it. What would the effect of a mutation be that would cause the G sub S alpha to stay on? What if it didn't even need a GEF? What if it was permanently in the switch position where it didn't need a GEF? That's dangerous, and that happens in that precocious puberty model. So it doesn't need luteinizing hormone to tell it to turn on. It's on all the time. Okay? And when it does that, typically it causes cancer of some sort, okay? But you can also think of other mutations. Mutations that prevent GTP, ACE from working. I would leave it on, okay. All right, so this protects against that kind of problem. This is called desensitization, or you actually use specific terms. You can use receptor sequestration is meaning you hide it, but you still have it available to use, okay? So if you do that, you use receptor-mediated endocytosis, which you now know frontwards and backwards because you're going to be quizzed over on Thursday, to store it in the early endosome, and then you just hold it there until you need it, right? And to get it to go back to the cell surface, will it go straight to the cell surface from the early? If this is the early endosome, where does it have to go first before it goes back to the cell surface? You're recycling this. It's got to go to the recycling endosome, and then it can go back to the cell surface by the process of exocytosis. And you know how that works too, right? Snares, B snares, T snares, and all that. So that can be. So this is a recycling pathway, but it can also temporarily hide the receptor from the ligand, so it's an off switch. Okay? So this is temporary. Now, if you have a really dangerous receptor, like the opiate receptor, what else do we talk? Growth factor receptors. A lot of times those are too dangerous to be recycling. And so what we do is we go to the early endosome, and then would you ever go straight to the lysosome? No. You would go where? The thing that takes all of that stuff is the late endosome, and then it becomes the lysosome, or fuses with the lysosome. Okay? So that's going to chop it up. And so you'd have to have transcription and translation to generate a new receptor. So that's receptor downregulation. That's what you call that. Okay? Now, can you inactivate the receptor? Absolutely. Okay? You can inactivate the receptor simply by phosphorylating the cytoplasmic domains of a G protein complex receptor. And the things that do that are called Greeks. Okay? G protein coupled receptor kinases, Greeks. Okay? And so that phosphorylates it in these domains, and that prevents pretty much the G protein from interacting with these domains that it needs, and so that shuts it off. And it will only do this if there's a ligand bound to the receptor. So only activated receptors get this treatment. Okay? Now, so that's a type of receptor inactivation. Okay? Now you can bind different inhibitory proteins like arrestin that we talked briefly about. So arrestin will then recognize that little subset of phosphorylation sites. Those are always serine threonine. This is not a tyrosine kinase. The off switch for this, or receptors mostly, is serine threonine phosphorylation. That tends to be an off switch for all receptors. Okay? And so it particularly is the case if this is the adrenergic receptor, this protein that binds is actually called beta arrestin because it binds to the beta adrenergic receptor. So that's where it was first discovered. But arrestin just sits there and it prevents any interaction with the G protein. You can see how there's no way a G protein is going to bump into this and get through this hodgepodge and be activated, right? But if you really want to be sure, arrestin will actually recruit AP2 and clathrin to this receptor and pull it off the cell membrane. And that will allow it to either undergo receptor sequestration or receptor downregulation. That's all mediated by arrestin recruiting the machinery to cause endocytosis. That's the common pathway for both downregulation and receptor sequestration. Okay, so we got through a lot of stuff. That's going to pretty much finish up the G protein signaling. I spent a lot of time on it, but because remember, for those of you that are going to go on to professional school, lots and lots of drugs do something somewhere in this pathway. Okay. All right, let's take a break. So we're looking for this, but caffeine is going to stimulate cytokine because it prevents its break. So that would be the answer. I said what you really need to do is you need something that turns on the possible. And now we know what we do is but that's what you would use. So yeah, be sure and understand if you're inhibiting the classical history, it's going to strike you. But that's exactly you want to turn on the crosstalk. As long as the oh, it didn't. So you got it. Oh, okay. So she popped that. Well, the real stopping place is okay. So it started signal. That's the end. There's no signal. What I don't even know which way. But they're stuff. Okay, thank you. Okay, why don't we get started again? I don't want you to take very long to look at this. In fact, it's just a really attendance thing. But just for your own edification, I'll put that up there. This is really straightforward. Thank you. The answer is C, by the way. So all I want you to do is click in. So just put C. Actually, you can click anything that's first 5 answers. But I'll let you look at that at your own time right now. But right now, just get out your clickers, and I'll give you about 30 seconds to do that. We'll move on. That's one of the things we're going to have to kind of cut down on. Oh, yes, like her. Just answer anything. Can she do it? Yay! Okay. Okay, wow, we got some smart people in here. You want to just do quiz 3 that way? Okay. All right. Now, so one of the things we want to answer is this question here. What makes the receptor tyrosine kinase family likely to be activated in cancer cells? And actually it's involved in diabetes and lots of stuff. So there's lots of diseases that make us want to understand this pathway really well. So a lot of time has been spent trying to figure out how it works. Okay, so here's the family itself. And what you notice about most of the family members, except for this really odd one, which is insulin receptor, is that most of them are monomeric, single-pass membrane proteins. You got one thing there, okay, and then they got these large extracellular domains which are responsible for identifying the ligand. Okay, so for example, our poster child for this is going to be, well, we have 2 actually. Epidermal growth factor is a very dangerous protein. It's overexpressed in lots of breast cancers and lots of different kinds of cancers, but it's a very potent growth factor. So the ligand binds to this, and its job is to dimerize 2 of these. So the general plan for most of this family is to take the receptor, and so here, that's just a table that shows all the various different members of the family, and we're going to only look at a couple. One of those being epidermal growth factor, okay? And so its job is to dimerize 2 of those receptors. And that's sort of the safety switch, is that unless this is dimerized, these 2 intracellular domains can't talk to one another. And the way they talk to one another is to autophosphorylate. They actually, instead of autophosphorylation, the appropriate term is to cross-phosphorylate. It doesn't autophosphorylate. In other words, this kinase domain doesn't reach up and phosphorylate itself like the CAMP kinase 2 did. Instead, it phosphorylates. This is sort of like the 3 Stooges slapping each other, right? Get it back and forth, okay? And so they load each other up with phosphates that are specifically on tyrosines. Tyrosine phosphorylation is generally a signal for cell proliferation. It's used for other things too, but it's a very specific thing. Okay, so you've got tyrosines that are phosphorylated. The reason that's important is that this creates a binding site. So the receptor itself has a couple of functions. Number one, it's an enzyme that's activated by a ligand. Okay. Number two, when you create these phosphotyrosines, those are binding sites to recruit other proteins. Now, the way you recruit proteins is specifically there's a domain on many proteins called an SH2 domain, and an SH2 domain it's like a little you can think of it as a what are the little building blocks? What am I trying to think of? Legos. It's a Lego, right? And it plugs in to these binding sites, if and only if it has an SH2 domain. Okay, and let me show you what that looks like. Okay, so you're gonna recruit these proteins, and so there's an SH2 domain there. There's an SH2 domain here. An SH2 domain is on the protein that's binding to the phosphotyrosine. Okay, you can think of it as being a phosphotyrosine receptor. That's a good way to think of an SH2 domain. Where does that word come from, SH2? Sarc homology 2 domain. It comes from our old friend C-Sarc, because C-Sarc is able to bind to phosphotyrosines. And when you recruit them all together, they talk to one another and they create these incredible signaling pathways that spin off of this. So the two functions of the receptor is to create binding sites that act as a scaffolding. And then to create those, you've got to be able to phosphorylate yourself. So it's got a kinase function and then a scaffolding function. Okay, so this is the molecular picture of an SH2 domain on a protein that's going to try to stick to the activated receptor. And notice it's got two pockets. It's got one pocket for phosphotyrosine, because that makes this protein a phosphotyrosine receptor. Right? And then it also has a little region that will help specifically recognize certain amino acids, because it won't bind to any phosphotyrosine. It's got to have, it's almost like a coincidence gate itself, but it's really not. Okay? It's a single plug-in that has two holes. One of those holes is for the phosphotyrosine. The other hole is for specific amino acids. You got to have both of this thing will not plug in. Okay? So it's very specific. And all kinds of different proteins will plug into this. We'll talk about this, but now you know what this is capable of doing. This is phosphatidylinositol 3 kinase. Remember, this is one of those RAB effectors. Okay, but in this case, it's not dealing with RAB. It's going to be close to the plasma membrane. It's going to phosphorylate the cytoplasmic leaflet and create binding sites for proteins. So this is this is a component of an entirely different signaling pathway that we're going to talk about. This is a GTPase activating protein, so this is going to turn off G protein signaling. Okay, we know what that's going to do. Phospholipase C gamma. Okay, this is the same enzyme as that phospholipase C beta, except it plugs into a different receptor. Instead of plugging into G protein coupled receptors, the gamma means that it plugs into a phosphorylated tyrosine residue on a growth factor receptor. What does it do? It does exactly the same thing. You don't have to learn a new pathway. It's going to split IP3, it's going to drop, you're going to have DAG in the membrane, it's going to activate. So this thing is going to activate the protein kinase C calcium signaling pathway that you already know about. Okay? So you can see how this is going to assemble a really bizarre array of stuff. So what we need to do is we need to look closely into the signaling pathway and break those pieces into smaller units. Okay? So the first pathway we're going to look at is the MAPK pathway, and it was identified in, strangely enough, Drosophila. So this is the compound I of Drosophila, and each one of these are called an ommatidium. Okay? And it contains various photoreceptive cells. And so to generate a fully active photoreceptor cell, it goes through development, and R8's the first one that develops, and it recruits R2, and that stuff is not important. But the last one that's added here is something called R7. It's receptor 7, photoreceptor cells. And it turned out in one of these poor unfortunate flies, there was a mutation where it was missing R7. So it looked like this. And so in their infinite wisdom, these Drosophila scientists called this 7-less. So this is 7-less, the mutation called 7-less. And what it turned out to be is that 7-less is an RTK. Okay, so the trade name for all those receptors that phosphorylate themselves on tyrosines are called receptor tyrosine kinases, or RTKs. That's what we'll call them. So this could be the epidermal growth factor, okay? Here's the ligand, could be epidermal growth factor, and it doesn't show it dimerizing, but you now know it's going to have to recruit a second brother, right, so that they can cross-phosphorylate. So this is a little inaccurate, but we're trying to, you've already learned that part, so we're not going to belabor it. The first thing that this protein recruits is it phosphorylates itself, because it's got another brother here that's dimerized, puts that phosphotyrosine on, and then it recruits this protein, GREB2. And since you're asking, that stands for growth factor receptor binding protein 2. That's why we call it GREB2, because we don't want to say that. All right, so GREB2 is a Lego, but it has binding parts to two different Legos on it. it's got an SH2 domain, which you now know is a phosphotyrosine receptor. And so it's going to plug in to the activated growth receptor, right? That's why it's called that. It's going to allow it to bind, and then it's, with its other LEGO exposed, it's a different LEGO. It's SH3. Now, if SH2 is a phosphotyrosine receptor, this must be a receptor for a different kind of protein. And it's a receptor for proteins that have lots of kinks in them. What makes kinks? Prolines. So it's looking for binding partners that have polyprolines. That's what an SH3 domain is a receptor for a protein like our old friend, well, it's not our old friend, this is RAS-GEF, okay? And we're actually finally we're going to learn what RAS does after all these years of talking about it. So this is RAS, and it's inactive. And so to make RAS active, you need its GEF, right? And so its GEF has polyprolines that allow it to be recruited to GREB2, which is stuck to the receptor. So the signaling is going like this, bringing the RAS-GEF to the cell surface. Why is that a good idea? If your job is to turn on RAS, why do you want it recruited to the membrane? Yeah, because RAS must be activated at the cytoplasmic leaflet of the cell membrane. So RAS is up here waiting, you've got to bring stuff to it, okay? And so this is a really exquisite control process because it can be controlled in any of these places. And so RAS-GEF is kind of clumsy. So the Drosophila people called it SOS, which stands for son of sevenless, okay? And so that's where the funny name comes from. Son of sevenless is bound to GREB, which is bound to sevenless. And of course the Drosophila people went further than that. The ligand that binds to sevenless is called BOS. Bride of sevenless. So you have bride of sevenless, and you have sevenless, and you have son of sevenless. Makes this hearty little family. Okay. The whole point of this family is to turn on RAS. Okay? Understand that this is not a kinase, that's not a kinase, and this is not a kinase. What kind of protein is this? That's RAS. You now know where to put that in the GTPase superfamily. It's a monomeric GTPase, okay? Or if you want to be not very careful, you could call it monomeric G proteins, but I don't like that term. Anyway, so this is RAS, and so the whole job of SOS is to bind to it. It's a GEF, so you know what it does. It shoots out the GDP and picks up GTP. Now, RAS is on, okay? But remember, RAS is not a kinase, okay? It's a GTPase. The thing that turns RAS off is a gap, and you remember that the gap is conveniently positioned on the membrane itself, on the receptor, so it's ready to turn it off as soon as it turns on. You do not want to leave RAS on. If you do, you get cancer. Okay, it's as simple as that. So the off switch is ready to turn RAS off very quickly after it turns on. Okay. All right. So now, how can you get the cell to proliferate? You have to talk to the nucleus. You have to go in and activate transcription factors that you're already familiar with, like Jun and Phos, okay? So how do we get from the plasma membrane into the nucleus? Well, you have to activate this triplet of enzymes. So now we're going to finally start talking about kinases, okay? The first kinase that's activated, which is activated by RAS, is, so the generic scheme is MAP kinase, 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 activates MAP kinase, kinase, which activates MAP kinase, which then does all this good stuff. Okay, but that's really clumsy, so we'll give it some specific names. A very specific family is, the first one's called RAF. Okay? That's the one that's activated by RAS. Not phosphorylated, it's a protein-protein interaction. RAS bumps into it, activates this kinase. What does it phosphorylate? It phosphorylates MEK. Okay? MEK, all right? MEK, also known as MAPKK, because MEK is going to phosphorylate the big daddy of them all, which is MAP kinase. We actually, the way MAP kinase got its name is that one of the targets that it phosphorylates are cytoskeletal proteins. So it was actually microtubule-associated protein kinase. That was one of its first activities. Guess why that would lead to cell proliferation? if you're phosphorylating microtubules, what do you need to do to microtubules if you want to divide? Well, what you really want to do is you want to break, so if they're all over the place, you want to break them apart and reconstruct them as a spindle fiber. And that's exactly what a dividing cell, either normal or abnormal, wants to do. So MAP phosphorylates many different things. But let's go back. What's the first question you always ask about a kinase? What class of stuff? Is it a lipid kinase or is it a protein kinase? Well, we know these are all protein kinases. This is a serine threonine. RAF is a serine threonine. Now, MEK is a little weird. We call it a dual specificity kinase because it phosphorylates serine threonine and tyrosine. That's because MAP is a little dangerous guy to be turning on. And so we actually have to put a couple of phosphates at two different sites. One's a tyrosine, I think the other is a serine. Okay? So this is actually a dual specificity kinase. Okay? MAP kinase is just Joe Blow serine threonine kinase. But it phosphorylates lots of targets that turn on translation and transcription to get the cell ready to go into mitosis. One of the things, of course, it has to do is it's got to go in through the nucleus. The reason it will not go into the nucleus before it's phosphorylated, so it has to be phosphorylated in order to dimerize, then it can go into the nucleus and phosphorylate transcription factors. Okay? And one of those transcription factors is an old friend of ours. So you have this pathway, RAS activates RAF, which activates MEK, which is phosphorylated, which activates MAPK. This is now in a phosphorylated state that it can dimerize. That's the secret. It's got a kind of like a steroid receptor. Steroid receptors need to dimerize, so does this. Okay? So then that allows it to expose a nuclear localization sequence, go in through the nuclear pore, and you know how all that works now. It's going to activate Jun and Phos. That's going to be one of those early transcription factors that's going to shove the cell into the cell cycle, which will be the topic of the last lecture. Okay? So just understand that this pathway turns on lots of genes that cause cell proliferation. That's all you need to know at this point. So you need to be able to put this pathway together very quickly in your mind and understand, you know, how does that act to that? How does that talk to this? How does that talk? Which ones are kinases? Which ones are protein? Which one have SH2 domains? What has SH3? That's the critical thing. But if you can just write this down, then you'll be familiar with the pathway. This is called the MAPK pathway. Okay? And it's one of the pathways that leads to cell proliferation. And the details are all written down here. Okay, so we've talked about all of that stuff. Now, that brings us to the question, we'd like to be able to answer this question, is why are cancer cells more likely to have this pathway mutated or activated? And it's pretty self-explanatory because these growth pathways use these RTKs in order to activate the MAPK pathway, which causes cell division. So that's one thing. If we can activate the MAPK pathway, typically we will cause cell division, and that's what cancer cells want to do. Okay, it regulates gene transcription, which is required to go into the cell cycle and go through it to cause mitosis. It causes this cascade of phosphorylation that activates lots of proteins that reorganize the cell from something that's differentiated into something that can go through mitosis. Okay. So let's talk about the results of any kind of mutations in this pathway. If it's a developmental pathway like that poor Drosophila, it's going to miss an eyeball, right? But in humans, it causes all kinds of developmental abnormalities. So it's usually, most mutations in these growth factors are not compatible with life, right? But also we get mutations that lead to cancer later on. And what we need to do is distinguish two different kinds of cancer-causing genes. And we're taking this opportunity to talk about this because the RTKs are one of the essential components of cancer, right? Typically, one of the cancer-causing genes that's overexpressed is RAS. RAS overexpression is not a mutation. Where do you think the mutation is? So if RAS is way overexpressed and the RAS protein is normal, where's the problem? That's a good guess. Upstream. Upstream from what?
Maybe,
That's actually a really, that's a good question. You started it off. Let somebody else. Good idea. So that's true. It's got to be upstream. So the protein's normal. So what controls RAS? The receptor. So maybe the receptor's overexpressed. In fact, this cancer problem that we were studying, which is cutaneous T-cell lymphoma, that's what one of the first things we found. The reason that they escaped the HDAC inhibitors, and those of you that went to the talk, you saw that person that had cutaneous T-cell lymphoma. It's really awful. And so you treat them with HDAC inhibitors that completely cleans them up. And it's amazing, except it lasts for 7 months, and then it comes back with a vengeance. And so what we did was to figure out what the resistance mechanism. Why do these people become resistant to the cancer treatment? And it turns out that the insulin receptor was overexpressed. And insulin, of course it regulates blood glucose, but it's a really darn good stimulator of cell division. And so that's what was allowing these people to escape the HDAC inhibition and go back into full-blown lymphoma, right? So we had to figure out what the problem was. And the first clue was the insulin receptor was overexpressed. So that's a good choice. Now, I'll tell you the rest of the story as we get further into this. But so yeah, upstream. But it could just be that the RAS gene, what controls the RAS gene at the level of transcription? Think back to quiz 1, because you're going to need to. RAS is a protein. How do you control protein? Transcription. Yeah, transcription factors. Could be mutated, so they're always sticking to the DNA. You could have nucleosomal problems, so that the nucleosome, the epigenetic changes have uncovered the RAS gene inappropriately all the time, so it's never hidden, it's always open, right? So you could have acetylation of the nucleosomes around that RAS gene that keeps it open and on and expressed. That would overexpress it. Or you could have some transcription factor that's left on. Let's say it could be the estrogen receptor that no longer needs estrogen. That's dangerous. Okay? So you can think of all these things. So this really starts to bring the entire course together about ways in which you cause cancer. So anything literally upstream could be any of those proteins in the pathway, or it could be the control over that protein itself. Also, it could be that the gene itself is mutated, so that RAS is messed up, so that RAS no longer needs sauce. RAS cannot break its own GTP. That would cause RAS to be really bad. Okay? So in that case, what you're doing is you're taking something, normal RAS, the structure of RAS, if it's normal, we call that a proto-oncogene. So there's absolutely nothing wrong with its structure. Proto means that it has the ability to cause cancer, that is oncogene, right? What makes it an oncogene is when it picks up a mutation, A, that either causes it to lose control over its activity, so it's always on, or B, there's some upregulation or mutation upstream that's overstimulating it. In both cases, RAS goes from being a proto-oncogene to a full-blown oncogene. Okay? Oncogenes are stimulating cell division, right? Now, if you have oncogenes, or you have things that stimulate cell division, you obviously have a different type of signal called tumor suppressors. So these are the nice off switches. What kind of off switch have we talked about in this BAT-K pathway? Just mention it briefly. Let's go back to it. We have about 3 minutes. Okay. Getting close. That's an off switch. Okay. So that's a tumor suppressor gene. Now, to get cancer, you need one of two things. Actually, you need probably both things to happen. You need to have oncogenes that turn on that cause overproliferation, and you have to inhibit tumor suppressor genes. So it's the inactivation or inactivating mutations or suppression of their transcription turns off the tumor suppressors. That will cause cancer. So it's not all about turning things on, it's also about suppressing the natural off switches. We just talked about all those off switches for the G protein coupled receptor, so we need to go a little bit further and describe how you turn this guy off. Okay. All right. Where does that bring us to? Okay, so we've talked about that. And so understand that I think we're going to pretty much stop there. When we come back on Tuesday, what we'll talk about is the last little member of this system, which is the insulin receptor. And so I'll talk about why is it important in cancer and why is it important in type 2 or diabetes in general.
You said the casein.
The which one? MAC?
Yeah.
Which is, MAP kinase, although I would never call it MAP KK.
Okay.
But just call it MAC.
Okay.
It's too complicated to call it that.
Okay, so you said the first one, the MAC. The first one is cesarean. The second one is the goal.